So I wanted to start this off by giving some rationale as to what we are doing with the GenRe-Mekong project and with the genetic report cards. Now, I will ask you to forgive me if I'm going to say things that you already know, but we have, I think, quite a wide variety of expertise here, and so I'm not going to assume very much, and I will just give you some rundown as to the motivations for doing our project. So the main drive was antimalarial drug resistance. So a few years ago, maybe around 2008, 2009, there started to be reports that in the greater Mekong sub-region, there were more and more parasites resistance to Artemisinin, which is the main drug that we use to treat malaria. So this threw us in a panic because in the past, other drugs have failed, and then this failure has become global, and then that set back the fight against malaria. So there was a lot of focus on eliminating malaria from the GMS, and this is part of where this project came from. Okay, so what is actually malaria drug resistance, antimalarial drug resistance? What actually happens that drugs are not working anymore? So from a clinical perspective, drug resistance means you try to treat the infection and the infection doesn't clear, right? And which means that the drug that you're using to kill the parasites is not working, it's losing efficacy. Now, the evidence in malaria, typically, what we do is we look at a blood smear after 3 days, and we can see whether there's still parasites in there, or else we look at whether there's a recrudescence. So we follow up, and after certain days, does the infection come back? And then you have a problem of asking yourself, well, is it the infection came back, or this person got infected by another mosquito? So we have to distinguish between the two. But so generally, when we see the numbers of these rising, then we worry, right? And ultimately, there are many reasons why a treatment may fail. There are some drugs, for instance, that require you to eat some fatty food in order to be properly absorbed. So actually, there could be other reasons why drugs fail. But when you have quite a few cases where the infections recurs, then you pretty much know that there is drug resistance. Now, why do malaria parasites become resistant to drugs? Usually, it's because something in their biology changes. So I'll give you an example here, which is that of chloroquine. So, the malaria parasites live in our red blood cells, right? And the way they feed on the hemoglobin that's inside the blood cells. But the hemoglobin contains iron compounds, and when it's broken down by the parasite, it becomes poisonous for the parasite itself. So it has to be converted in hemozoin, which is less poisonous for the parasite. Now, what chloroquine does is it stops this conversion, so essentially it poisons the parasite. So parasite eats and dies from the byproducts. So chloroquine, very good drug, right? In the 1950s, it was used all over the world to kill the malaria parasites. But in falciparum, what actually happened is that there is this protein called CRT, which is it's like a pipe out of the digestive vacuole, which is like the stomach of the parasite. And this CRT normally doesn't let chloroquine out. But what happened is it got some mutation such that it changed the shape of the channel such that it could flush out the chloroquine. And then the parasite wasn't getting poisoned anymore and chloroquine was useless. Okay, so this is the type of changes that we are looking for. And how does that protein change shape? It's because of some genetic changes. So proteins have, they're like little biological machines inside an organism, and their shape, their function is determined by the DNA that produces them. And if the DNA changes, then the shape changes and the function changes. And this is, so usually, in order to confirm that some parasites are resistant, what we do is we test them in the lab. If we can culture them, we keep them going for like 48 hours, and we add different concentrations of the drugs, and we map out what drugs do they get, and what concentration do they get killed. And what happens is, as they get resistant, you get higher and higher concentration are needed. So basically, we can test them. In some cases, we find it very hard to test the parasites. For example, Plasmodium vivax is a disaster because we cannot culture it. It doesn't lend itself to culturing. So that's the reason why, for instance, we don't know what mutations make it resistant to chloroquine. So now, why is drug resistance a big problem? Because not only does it stop the drug working in a particular place, but those parasites, they spread. And in some cases, they can spread very far. So when chloroquine resistance started, about 1957 was the first reported case, which was at the border between Thailand and Cambodia, more or less. At that time, it started spreading because chloroquine was used absolutely everywhere. So those parasites were very strong, not only in Thailand and Cambodia, but also in other countries that were using chloroquine, right? So they started spreading, and they probably spread through India into Africa. And actually, they made chloroquine useless pretty much all over the world. And so nowadays, we still use chloroquine, but only for Plasmodium vivax. This is falciparum. Now, later on in the 60s, another drug, in fact, a combination, sulfadoxine pyrimethamine, again, we started getting some resistance, again, pretty much in this region, probably Thailand and Cambodia is the epicenter. And again, it spread across, and it went to Africa, and SP also got reduced effect. And mefloquine, we also started getting resistance in this region in the Eastern GMS. And now our main antimalarials are what we call ACTs, Artemisinin combination therapies, which are made up of two drugs. There's artemisinin and a partner drug, right? Artemisinin is a fantastic drug. It kills parasites with incredible speed. So you take artemisinin, your parasite load immediately goes down very quickly. But artemisinin only lasts a very short time in your bloodstream. It's metabolized very quickly. So what we need is another drug, which needs to complete the job of artemisinin. And actually, there's a variety: piperacin, mefloquine, pyronaridine, etc. And now in the GMS, different countries may use different partner drugs. So, for example, in Laos, use lumefantrine. In Thailand, now in the northeast Thailand, use pyronaridine. In Vietnam also. And in Cambodia, mefloquine. And these combinations are working very well as long as there is no resistance to the partner drug, because we do have resistance to artemisinin. But if the partner drug is working, we're still not getting failure. But the problem is, in the past few years, the countries in the region were mostly using piperaquine as a partner drug, and resistance to piperaquine emerged, and it was a bit of a disaster. So now all the countries have changed. And actually, our job was to inform the control programs about where this resistance was located. Okay, so this has been largely our main mission recently. Now, you know, there is something about the parasites in this region that have made it that first chloroquine, sulfadoxine, then mefloquine, and artemisinin resistance all emerged here. So basically what WHO decided 10, 15 years ago was better completely eliminate as high priority from this region, because there is something about these parasites. Maybe they adapt better, something like that. So we want to stop this from spreading, especially spreading to Africa, because obviously that's where most victims of malaria are. So this is where this project really came from, in order to try to stop the spread. Now, how does the spread happen? Well, interestingly, the spread of drug resistance is really driven by us. It's really driven by our attempt to eliminate. The more we try to eliminate, the more pressure we put on the parasites to become resistant. Okay, and actually, it's quite straightforward if you think about it. That, let's say you have a population of parasites that are infecting people, and let's say that most of them are low resistant. You know, they're sort of yellow and a little bit orange there, but there is just a very, very few parasites that are resistant, right? So now this population infects people. People go to the clinic and they take antimalarial, right? Antimalarial is more likely to kill the yellow ones than the red ones, right? So what is going to happen is you just left with these guys. Now these guys are going to now multiply, right? Because that's what they do. So you now get a bit more red than before, you see? And then now these people who are infected, they go back to the clinic and they get more drugs. So, and again, it's mostly the yellow and orange ones that get killed. So there's more and more red ones left. And this continues. And very quickly, you get to a situation where the more we give drugs to people, the more we get resistant parasites. And it's not because we're doing things badly. It's really because this is the natural way in which the parasites respond. So what can we do? What can we do? We can change, for example, the drugs we're using. When a parasite population becomes resistant to, let's say, piperaquine, we stop using piperaquine. We use mefloquine in the case of Cambodia, for example. But then we have to monitor the population in case it develops resistance to mefloquine. So that is one thing that we can do. By the way, it's very expensive for a country to change frontline treatment. I'm sure we can ask all the NMCP representative here. It's a very big headache. You only do it if you are really forced to, because you may end up with a very big storeroom full of drugs that you have to throw away. So, and also we don't have an infinite number of antimalarials. Once you run out of piperacin or mefloquine, then you don't know what to use next. So we have to be very, very careful how we do this. And of course, preventing this from happening is another another way, and several strategies have been proposed. For example, using triple combinations, where instead of having one partner drug to rescue, you have two, so they protect each other, sort of thing. So these are different ways of approaching the problem. From our viewpoint, what we like to do is to have some genetic markers that tell us whether a parasite is likely to be resistant, and to monitor that and to put it on a map. And in that way, we can actually inform the control programs so that they can take these decisions more with more knowledge in hand. Okay, so what can we monitor? Well, basically, there are genetic mutations that we can observe and that we know that are related to drug resistance. So when you have, as I explained earlier on, you have mutations in the DNA. The DNA generates a protein, so the protein structure changes as a result of the mutation, and therefore the protein works in a different way, and this actually changes the response of the parasite to the drug. Okay, and this can lead to treatment failure, as it were. And how do we know this mutation? Actually, it's really hard. So what you need to do is to take a lot of parasites that are resistant and a lot of parasites that are sensitive, and you need to get the genetic sequences, and you need to compare the two groups and actually find, for a particular at a particular location in the DNA, there's a different nucleotide in the two groups. But in order to get this type of result with statistical significance, we need hundreds, if not thousands, of samples. And as I said, in some cases, we can we can culture the samples in the lab so they can be tested, because we need to figure out, is this parasite sensitive or not? In some cases, we can get from clinical cases, so we know that if they are resistant or not. But generally, it's a hard problem to solve. And actually, we don't have mutations that tell us about resistance for every drug, only for some drugs. By the way, I would like to point out one very important thing here. You can only know that there is a mutation that makes parasites resistant only if there are resistant parasites. You cannot guess what mutation is going to make them resistant. So, sometimes now I am asked the question, do we have a marker for paranaridin resistance? And my answer is, do we have paranaridin resistance? Because if we don't, then we cannot have a marker. We don't know. Sometimes in the lab, you take parasites and you subject them to the drug to see if they develop resistance. And then you try to find out. But actually, that only tells you what happens in the lab. It may not happen when they infect people. So sometimes we don't have a marker for resistance, and sometimes it's because it's for good reasons. A very interesting case is lumefantrine that you use in Laos. Now, lumefantrine has been used a long time. It's used a lot in Africa, and we don't know if there is resistance to lumefantrine. Some parasites have different sensitivity, but difficult to know. So actually, even though a lot of the world is using this drug, we don't have a marker for resistance. It will come. Okay, I want to give now a little bit of, spend a few words to tell you about the project and about how we operate. Some of you, of course, are very familiar with that because you work in the operations of this project. But the main idea is that we work with national malaria control programs, and we ask them to decide on a sampling strategy where we're going to get blood samples from. And we set up what we might call a local sampling network. So there are usually country coordinators that work with the clinics, with the health centers and so on, in order to collect samples from symptomatic patients. And these samples are simple to collect. They're dried blood spots, and they get sent to the laboratory for processing. And actually, we have here representatives from 2 laboratories that work with us in Vietnam and in Thailand. And the processing of the samples gives a lot of readouts of genetic mutations that are likely to be associated with drug resistance, and then other information about the genetics of the parasite. And what we do with those results, those genotypes, we translate them into maps and into reports, and we give this back to the control program. And then we just keep on going around. And every time the situation, the epidemiological situation may change. So you may change the sampling network or sampling strategy, and you may give different information and so on. So hopefully we keep on circling until malaria is no more. So the basic technology underneath it all is spot malaria. And I'm paying homage here to the person whose idea it was, and he died only a few days ago, and Dominic Karkowski. So he was the person who really imagined all this thing happening. So thank you very much, Dominic. Basically, the idea was to use some leading edge sequencing technologies in order to make the most of very, very small blood samples. The idea was that we were going to have some SOP for collecting samples that could be integrated in the operations of even the simplest health center in the remotest places. So the idea is to make it very simple, collect a small sample, and then use all the technology you have to extract as much information as possible. Okay, so new laboratory methods in order to process these samples, and finally deliver this information in a meaningful way for the public health officers. Now, surprisingly, we knew how to do the first 3 things much more than the last one. It's for us technologists, getting a series of mutations was okay. You know, we just had to get ourselves organized. But actually translating them into things that are meaningful and actionable for public health, that's a lot trickier, because you have to transform and you have to interpret the data. So this is really what we are going to be covering in this particular course. And just to quickly go through how we integrate with the different entities. So yes, we work with the NMCPs on the strategy. We work with local coordinators, and there's quite a few of you here, that sort of work with the control programs in order to manage the samples, to collect them, to aggregate them, to make sure that they're accompanied by quality data and so on. Then we work with the laboratories and our scientific partners within the countries and regionally. And I should point out, Dr. Nguyen is right at the end there, and she runs the laboratory in Ho Chi Minh City that processes samples from Vietnam, Cambodia, and Laos. And actually, new laboratory is being set up, and we got some representative from there here, well, down the road here at Mahidol University, which is run by Professor Malika, and they will be processing the Thailand samples. And once the samples are processed, there's a lot of computing that has to be done in order for us to know what the mutations are, and that's done by informatics pipelines that are in the clouds that are run at the Sanger Institute, and we got Christina down there representing this part of the process. Okay, and finally, the reports and the maps are generally produced by my team here, and also in collaboration with the scientific partners within the countries. And the objective of this course is actually to extend this group now, so that more people can do that, more people can make use of the data. This is what the data looks like when you receive it in your email. And you might, unless you are sort of strong in malaria genetics, this really means very little to begin with, right? There's a lot of amino acids down here. There's a lot of there's haplotypes down here. There's genetic barcodes here. There's some easier to understand information, like where the samples come from, which part of the country. This one is quite easy. For each drug, it tells you, is it resistant or is it sensitive? Okay, so this is a good starting point. So you can see here, we have a rather problematic set of samples that are resistant to a lot of drugs. Okay, but we have some light at the end of the tunnel. You can see mefloquine is good to use here, right? So this is the type of thing that you will do today, sort of have a look at the raw data that we returned this morning. But actually, what is more useful is when we can create maps that actually tell us, has resistance spread where, and the difference between different years, perhaps, or how the situation is changing over time. So this is the type of reports that we send back to the control programs. This is an overview of the spread of resistance in different provinces, for instance. And then we also sort of create some infographics and things that can be put in presentations, etc. So the idea, so in order to create these, we use the GRC malaria library that you're going to be learning about in this course. And so just to underline that the course is really about this part, about what comes out of the genotyping process, the genetic report cards, and how to turn them into more useful stuff. Okay, and with this, I'll pass you on to Varanya, who will tell you a bit more about what happens in the rest of the day.